Hi guys, we're going to finish up chapter seven with talking about translation. Okay, so I'm going to bring up my PowerPoint and we're going to start talking about translation. So last time we talked all about transcription, which was how you use a DNA template to make a molecule of mRNA. So we used this template strand of DNA, did complementary base pairing. Thymine pairs with adenine, cytosine pairs with guanine. Adenine pairs with uracil in a molecule of mRNA, et cetera, et cetera, et cetera. Okay, so you've got this molecule of mRNA. You went through your process of post-transcriptional modifications, added your GCAP, your poly A tail, and went through splicing where you took out your introns. And now this molecule, mature molecule of mRNA, has been transported into the cytoplasm and we're ready for translation. In translation, you're going to use this template strand of mRNA to build a fully functional protein, okay? So unlike translation, where you had RNA polymerase enzyme reading these nucleotides one at a time and complementary base pairing, in translation, we're not reading them one at a time anymore. We're reading them 3 at a time, okay? 3 nucleotides on an mRNA strand is called a codon. So think of this as like we're reading words now instead of reading letters. Once you have something in English, do you read every single letter when you're reading a book? No. Letters can be misspelled and you can still understand the word. Words can be misspelled and you still understand what the word is. We're reading codons during translation, which are these sequences of 3 nucleotides, each of which encodes a single amino acid. So here, AAG is going to encode lysine, UCA encodes serine, GUC encodes valine, and you're going to all right, so you've got these one after the other, right? So if this encodes lysine and the next one encodes serine, you're gonna lysine, bring in serine and link them together. So notice they're all linked together. You're building this polypeptide before you can make this fully functional protein. All right, here is your genetic code. And your genetic code is basically showing you which mRNA codons correspond to which amino acids. Now, how do you know that this is all mRNA codons instead of DNA codons? How do you know it's not DNA? How do you know it's mRNA? Do you have any thymine at all in this chart? No, everything's everything's uracil, right? There's no thymine in this genetic code. So we're dealing with mRNA codons, and I want you to notice that each mRNA codon, some of them will encode a single, sorry, I'm moving my thing around. Some of them will encode a single amino acid like tryptophan, okay, with UGG encoding tryptophan, but a lot of them are going to be redundant, meaning like leucine, for instance, you've got 2 here and another 4 mRNA codons here. All 6 of them encode the same amino acid. That's a good thing. This is redundancy in the genetic code. It means that even if there is a mutation that causes UUG to be changed into CUG that encodes the same amino acid. So even though your DNA and then your mRNA changed, it's not going to change your amino acid sequence. That's a beautiful thing with the genetic code. 2 other things that I want you to notice are the things that are colored. Okay, so in yellow, you have AUG, which encodes methionine. Okay, AUG encodes methionine. This is your initiation codon. We're going to call it your start codon. Every single polypeptide is going to start with methionine. Every single one, you're starting with AUG. Okay, translation, you're going to have the ribosome. It's moving along until it sees AUG. If you have codons before AUG, they don't matter. They don't matter because AUG is your starting line. Then you have in pink your termination codons, meaning your stop codons. Now, UAA, UAG, and UGA, they encode stop. Is stop an amino acid? No. So do you include stop in a polypeptide chain? No. So let's do one together. Let's do one together. I want you to write this down as I'm calling it out. Get a pen, pause this until you have a pen. I want to do G, U, A, U, G, C, C, G, C, U, G, U, G, A. Okay, so one more time. I've got G, U, A, U, G, C, C, G, C, U, G, U, G, A. All right, now you'd look at this and you would say, I'm going to stop sharing for just a second. You'd look at this at yours, really, okay? And you'd say, all right, well, our first codon is GUA. Is that true? No, what's the first thing you're going to look for? AUG. The first thing you're always going to look for is AUG, that codon. So this is your first codon. Well, does it matter that you've got two nucleotides before AUG? No, it doesn't matter at all. They're not going to do anything. They're not going to be part of this protein, and that's totally fine. So AUG encodes MET. It is a real amino acid, so we're putting it in our polypeptide. Now I'm going to go back, and I want you to go through this. Our next codon is CCG. So you're going to find CCG. Your first letter is C right here. Your second letter is C, and then you find CCG right here. So your second amino acid is pro. For proline, this codon encodes proline. CUG is right here. That encodes leucine. LEU. And then UGA is a stop codon. Are you going to put, I'm stopping this again, are you going to put stop right here? No, because stop is not an amino acid. So your entire polypeptide is MET pro leu, and it's going to detach from your mRNA and go fold into its three-dimensional structure and make a fully functional protein with just this sequence, just with 3 amino acids, okay? That's how you do that. Every single time, the first thing you're gonna look for is AUG, and then you stop when you find a stop codon, okay? You don't need to put stop. Okay. You will have this available to you. Well, so you have this available to you anyway because it's an open book situation, right? So just make sure that you have a picture of this, or make sure you have this PowerPoint open when you are taking this exam. Now, the way that it works, that you have a ribosome reading any of these codons and then pairing it with an amino acid, is that you have a transfer RNA molecule, okay? One side of this transfer RNA molecule is called an anticodon. It's called an anticodon because it holds the complementary sequence to that mRNA codon. So this anticodon, this side will bind to your mRNA codon, and then the other side is the amino acid binding site. It's holding the exact amino acid that you need, okay? So for instance, if you had AUG, your mRNA codon was AUG, what's the complementary sequence to AUG? Well, A pairs with U in RNA, U pairs with A in RNA, G pairs with C. Okay, so UAC would be the sequence that's on this side of your tRNA. And then on the other side, you're holding on to whatever this mRNA codon encodes. So you would be holding on to methionine. Remember, that was our start codon. It encodes methionine. Commit that to memory, please. All right, 3 steps during translation. Same 3 steps as transcription: initiation, elongation, and termination. And they happen all at ribosomes in the cytoplasm. So during initiation, what's going to happen is that you have this small ribosomal subunit. It's half of the ribosome. It is going to bind onto what's called a leader sequence. Okay, binds onto the leader sequence just like the promoter or the TATA box in transcription. Binds to this leader sequence, and it's going to move along this strand of mRNA until it finds the start codon. Okay, it just so happened in this case it was really close by. Sometimes it would be all the way down here. It doesn't matter where it is. Here's another one. Okay, you could have a situation where instead of starting with this one, you start with this AUG. This happens all the time, and that's fine. Now, small ribosomal subunit binds to the leader sequence, moves along this mRNA molecule until it finds your start codon. A tRNA, transfer RNA molecule, on one end, it has your anticodon, and on the other, this is what we were just talking about, and on the other end, you've got the amino acid that this codon encodes. So one end, you've got the complementary base pair to this codon, and on the other end of this tRNA, it's holding on to your amino acid that you need, okay? So tRNA binds to the mRNA codon right here at the anticodon, and then down here, it binds to the corresponding amino acid. We already went through this. If you need this, copy this down, please. All right, tRNA, complementary base pairs to the mRNA. tRNA already carries an amino acid, is literally already holding on to the amino acid that you need to start building this polypeptide. Next, I want you to see, going from here to here, you had this small ribosomal subunit, and because this tRNA bound to AUG, you're going to have this large ribosomal subunit come in and close it all, okay? So imagine this is like an iceberg sort of situation, okay? It encloses itself in, but it's open on the sides. So tRNA molecules and amino acids are going to be able to get into this ribosome, this ribosome, okay? But it looks like it's closed. All right, it's enclosed. It already read AUG, and it already paired that tRNA. So what's going to happen? Small ribosomal subunit binds to the leader sequence, moves along until it finds AUG. When it finds AUG, tRNA comes, binds to it. A large ribosomal subunit comes and binds to the small to enclose this ribosome. It says, okay, we're ready for business. We are going to actually translate this mRNA molecule into a protein. Then this whole unit moves over one codon. So it's read AUG. Now it's going to move over to the next 3 to read this next word, this next codon. So here you've got GGA. You're going to have the transfer RNA that can bind with GGA, come into the ribosome, bind to it. On the other end, it is holding the exact amino acid encoded by GGA. Okay, so GGA, let's go look at this for a second. GGA is right here. It encodes glycine. So look at that. It encodes glycine, your tRNA bound to your mRNA codon and is holding the exact amino acid that you need. Before the ribosome complex moves over again, you have to bind your amino acids together. This is the whole point because you're building a polypeptide. So then it's going to move over. It read 3, moved over, read another 3, bring in the tRNA, bind your polypeptide, move over another 3, and when it moves over another 3, it's going to kick out this first tRNA to make room for a new tRNA, because it already read this, has no need for this tRNA anymore because you've bound methionine and glycine. So it moved over another one. You bring in a new tRNA that is holding the corresponding amino acid of UGU, which is cysteine. You bind together glycine and cysteine to make this polypeptide, and then it moves over another 3. When it moves over another 3, it'll read AAG, it'll kick out this tRNA because it's not needed anymore, bring in this tRNA that's holding on to lysine, you would bind these two together, moves over again, brings in a tRNA, kicks out the first tRNA, bind the polypeptide. Just over and over and over again. You're just elongating the strand of your polypeptide, just one codon at a time, basically. So inside, what's important is that inside of your ribosome complex, you have to have 2 of these tRNAs at a time. So even though you read this one and you brought this tRNA in, and you moved over to read this next tRNA, you can't get rid of this one yet until you bind those 2 amino acids together, because that's the entire point. Okay, that's the whole entire point. So you bind those 2 together. So at the same time, you can only have 2 things inside of the ribosome. Now, once you get to a stop codon, instead of a tRNA, a release factor protein is going to come into the ribosome. And what do you think a release factor protein does? It releases everything, okay? The ribosome subunits are going to detach. This polypeptide is going to be free floating now in the cytoplasm. It's going to go ahead and fold into this functional protein. And then you're done. You're done with translation. After you hit that stop codon, release factor protein comes, releases everything. mRNA will degrade. Polypeptide will fold into a functional protein. What are the steps of translation? Initiation, elongation, termination. And where in the cell does translation occur in the cytoplasm? At a ribosome in the cytoplasm. Now, protein synthesis, making a protein, going from DNA to RNA to protein, it takes a lot of time, it takes a lot of energy. So cells save energy by only producing the proteins that they need. Only producing the proteins that they need. This is like a summary image. We're not going over this, but you should test yourself with this. Okay, please test yourself with this. Guys, this is also for you. DNA is found where? In the nucleus. What process does it undergo? DNA to make RNA? Transcription. RNA is then modified. What are your post-transcriptional modifications? Or your post-transcriptional modifications? Adding a GCAP, a poly-A tail, and going through splicing. RNA is then moved to a ribosome in the cytoplasm. At a ribosome in the cytoplasm is where translation occurs, turning mRNA into a polypeptide and then a protein. Okay. Guys, we're going to talk just for a few minutes about mutations. So not all mutations are bad, not at all. You've got a couple of different types of mutations. You can have a substitution mutation where one nucleotide is replaced with a different nucleotide, or you can have what are called indel mutations, insertion or deletion mutations, and these cause a frame shift. So you'll see what I mean by a frame shift in just a second, but essentially one with 2, or 1 with 3, you have just shifted the entire thing. You're not going to be making the same protein anymore. So errors can be fixed, but not if mutations, excuse me, but the mutations are going to vary in effect. Okay, so some are going to be completely insignificant. That's important, and that's why in your genetic code, you have so much redundancy. You had 6 mRNA codons that would encode the same amino acid. Okay, so some mutations are going to be insignificant, some are going to be negative, some are going to be lethal. Most lethal mutations are going to happen before you are born, okay, where like the embryo or the fetus is not viable anymore. And then some are positive. And this is how, with random mutations, sometimes these positive mutations will lead to new adaptations in a species that will allow this individual with a positive mutation to reproduce, pass on that mutation, and survive better. All right, so your normal sequence is dog, 8, bat, and hat. Your codons are dog, 8, bat, and hat. Substitution mutation, instead of dog, 8, bat, and hat, you have dog, 8, cat, and hat. Notice, these are more or less the same words. You have the same number of letters and everything like that. You just had one of your codons change. So instead of bat, we've got cat. That was a substitution mutation. In a frame shift mutation, you go from dog, 8, bat, and hat, and you have a deletion. A deletion is going to completely change your codons. So it becomes this, this sequence, and here are your codons. That says something completely different than this, okay? Now, what's good, because we have start codons and stop codons, if this doesn't have a start codon, or if it doesn't have a stop codon, it won't be transcribed, okay? All right, so here we have a very common substitution mutation where in your DNA, so blue is your DNA sequence, in your DNA, you go from a is substituted with an adenine, okay? And this is going to change, instead of GAG mRNA codon, it's going to create a GUG mRNA codon. That's going to change in this chain peptide amino acid sequence. That's going to mean in sickle cell disease, normal red blood cells have this like random distribution of hemoglobin molecules. But in sickle cell disease, what happens is your hemoglobin levels are normally distributed. They stick to each other, which means they're not able to carry as much oxygen because they don't have as much surface area as they did in this random distribution. So you get sickle cell disease. It's going to change the shape of your red blood cells, but mostly it's going to involve this hemoglobin, okay? You get sickle cell anemia. Are mutations always bad? No, absolutely not. Mutations are not always bad. They create new variations in genes. Natural selection selects for traits that are favorable. Over time, you're going to have more diversity. You're going to have lots of genetic variation thanks to mutations. So sometimes they're bad, but we can thank mutation for the evolution of every different species, okay? Geneticists use mutations, plant breeders use mutations, et cetera, et cetera. There are lots of cool ways that we can actually use mutations to our benefit and to the benefit of, you know, different species looking to, not that you can wish into existence new mutations, okay? In a lab you can, but in nature, just acting on the variation that's already present in an environment. So mutations, they can be good, they can be bad, you know. All right, guys, that is all for chapter 7. And I hope you have a great week, and I'll see you next week. Next week, we're talking, well, you'll see it in the video. You'll see it soon. Bye.